Um, my name is Hannah Bui, and um, my partners for this video are Rohan Patel and Isabelle Hanson. Our project is Wild for Wild Rice, genetically modifying wild rice for increased acidity tolerance.
So recently, the boundary waters in northern Minnesota have been threatened by sulfate watersheds from sulfide or copper mining. Wild rice, the state grain, is predominantly grown in the area. The effects of sulfate have been shown to have a direct impact on the occurrence of wild rice. Studies conducted over a 5-year period show that wild rice populations becoming extinct with exposure to 250 milligrams of sulfate per liter or greater in tank water. Within the soil, acetic acid and sulfate react to reduce sulfate. One of the significant products from this anaerobic bacterial-led process is hydrogen sulfide. An increase in levels of hydrogen sulfide in the soil ultimately leads to a decrease in pH.
This is our proposed solution. Researchers recently created transgenic petunias that showed tolerance to acidic environments by inserting the MYB1 gene found in Raphanus sativus radishes. Our proposal is to insert the MYB1 gene along with the 35S promoter into wild rice genomes and to eventually introduce the transgenic plants into the wild. Inserting this gene into species of wild rice may protect them from increased water and soil acidity due to mining. The protection of wild rice is important given their cultural and economical significance in Minnesota. Since radishes, our source of the MYB1 gene, and wild rice are closely related, transferring the gene between them should be possible without many complications.
The MYB1 gene encodes an R2-R3 MYB1 transcription factor within the cell nucleus, which regulates the production of the pigment and anthocyanin. This controls the red root color phenotype seen in the radishes. Its overexpression in petunias and tobacco plants strongly enhances anthocyanin production, which ultimately enables tolerance to low pH stress. This picture is the chemical structure of anthocyanin, and the 35S promoter. We will be using the 35S promoter, and this is from the cauliflower mosaic virus, which enhances gene expression at a high frequency across all plant cell types.
So with isolating the gene and promoter and inserting it into the plasmid, the first step is that the MYB1 gene will be isolated from the genome of Raphanus sativus, also known as the radish, which will be obtained from cub foods using PCR with the below, forward, and reverse primers. The forward primer from the 5 to 3 end can be seen above, and the restriction enzyme that cooperates with that strand is SAL1. Reverse primer would be E. coli restriction enzyme. And the second step would be that the 35S promoter will be isolated from the PGFP GUS+, which is obtained from add gene using PCR with the below, forward, and reverse primers. And then the corresponding forward primer and then the reverse primers are listed below. And then you can see that the HIND3 restriction enzyme corresponds with that strand there, and then the SAL1 over there. And then the third step would be that restriction enzymes HIND3 and SAL1 will be used on the 35S promoter to insert it into PBIN19. Restriction enzymes SAL1 and E. coli, which will be obtained from Thermo Fisher, will then be used to insert the MYB1 gene into the PBIN19 TI plasmid.
This shows a diagram of our vector, which is the TI plasmid, and a better way to show the restriction enzymes. It contains virulence genes, which enable a copy of a segment, tDNA, of the TI plasmid to be transferred into plant cells, where it is integrated into the plant genome. CAN-R acts as a selectable marker by making transformed bacteria resistant to kanamycin.
Our vector diagram shows the gene and the places where the restriction enzymes will cut. To insert our plasmid into agrobacterium and test for successful insertion, the transformed PBIN19TI plasmid will be inserted into agrobacterium using electroporation. Then the antibiotic kanamycin will be used to select agrobacterium that have integrated the plasmid. To identify the cells that have taken up the transformed plasmid, the DNA sequencing will be used, the forward primer and the reverse primer.
So in order to transfer the plasmid into a wild rice plant and test the acidity tolerance, these following steps will be taken. So the cells will be collected from the plant by hole punching a leaf. Samples will then be exposed to the agrobacterium with the transformed plasmid. Next, the cells will be allowed to regenerate a new plant containing plasmid. The genome will then be sequenced to determine the gene's presence. 500 samples of the transgenic plant will be compared to a wild-type control group of 500 plants in a substrate with an increased level of sulfide. Molecular sulfide levels and plant survival rates will be measured daily throughout an entire growing season to test the plant's tolerance to acidity.
There are many other plants that are closely related to the Raphanus sativus. They include field mustard, cabbage, and garlic. They all contain the MYB1 gene, just like the Raphanus, meaning they are alternative sources for the gene. Radishes and wild rice both belong to the class Magnoliospida and diverge in the superorder hierarchy, with radishes belonging to Rosenae and wild rice to Lilianae. The relatedness of the two species increases the chance of a successful transfer of the MYB1 gene. And this is the phylogenetic tree on the right that shows the relationship to Raphanus sativus at the bottom. Possible evolutionary consequences. Some risks of wild rice hybrids being produced through cross-pollination of the genetically modified plant with other species in surrounding area. These hybrids may have unknown impacts on the species and environment.
So part of our ethics is considering the viewpoints of tribal nations in Minnesota. Manoomin or Pisan, which is the Native American term for wild rice, is more than just a crop or source of food to the Native American tribes that live in Minnesota. It represents their journey in migration, relationship to the land, and also represents their identity. It holds a value economically for many of the tribal nations in Minnesota. The alteration of wild rice in any form is not accepted within tribal communities. They believe that humans should try to connect with the land and learn from what the land is telling them rather than try and manipulate and control the natural forms of life. The tribal nations view the wild rice as elders who care and teach the community by conveying the message of the land and talking to the people. Some ways to work together would be to working alongside tribal members in order to respect their cultural and spiritual needs. Also, we could try and learn the ways of knowing of the tribes to better understand the importance of Manoomin. We could also respect the treaties and recognize the rights of Native American tribes that have already been in place.
Some other potential issues and considerations to take into account are the transgenic wild rice may lead to an increase in uptake of hydrogen sulfide, which can be consumed by humans and other organisms. People may have respiratory and neurological problems if exposed to higher concentrations of hydrogen sulfide, at least 100 times higher than typical environmental levels. Given that transgenic wild rice would be better suited to the increasingly acidic environment, it may have a selective advantage over other native plants. There wouldn't be a need to genetically modify wild rice if there were increased regulations or bans against sulfur or copper mining. Unfortunately, these efforts, even with scientific evidence, have not yet been successful. Some potential solutions could be that hydrogen sulfide concentrations within transgenic wild rice grown with various levels of sulfide can be tested to ensure that the levels are within the safe range. If the plants are above the safe range, then they will not be released into the wild. And also another thing is the passing of the Boundary Waters Wilderness Protection and Pollution Prevention Act, which was most recently introduced to the House in January 14, 2020.
The following are possible extensions of this project. 1. We could incorporate genes into another affected plant. Wild rice is not the only plant that is affected by boundary water acidification. Rooted aquatic vegetation, such as seaweed, are a vital part of the ecosystem and do not thrive in high acidity environments. The MYB1 gene could be inserted into other aquatic plants using agrobacterium to solve this problem. We could cross-pollinate to transfer genes to other plants. Other species that are in the same family as wild rice, such as water millet, could potentially be cross-pollinated with wild rice to create hybrids that are resistant to acidity. We could also use the MYB1 gene from other sources. The MYB1 gene is present in a range of different plants, which mean there are sources which could possibly be more closely related to the wild rice, seaweed, or watermelon. Being more closely related can lead to a better expression of the gene altogether.
This is the end of our presentation. These are some of the references that we used to make this presentation. Thank you.